thank the organizers for giving me this opportunity to present this work, which is published recently. So the talk is about using plant-derived microRNAs to target SARS-CoV-2. I work in this institute called the Central Food Technological Research Institute in Mysore, India, in the Department of Molecular Nutrition. So I think we may not need much of introduction about SARS-CoV-2. The previous speaker gave a very nice introduction about SARS-CoV-2 and what are the proteins that is present in that. So I'll probably skip that. But what is important to note here is that SARS-CoV-2 is a positive sense RNA virus, which means the viral mRNA is viral genomic RNA is like an mRNA. So once it goes inside the viral, inside the cells, the genome is translated immediately. Then you have lots of subgenomic RNAs as with, complementary RNA of the genome, which packages the virus, which makes the viral proteins and package them, sends them out. So I think now we have some specific treatment like vaccines, but at least probably 3-4 months ago, we didn't have vaccines also, and we had kind of mostly non-specific treatments or empirical treatments based on previous things. So here, what I would like to propose, okay, so before that, I work on this institute. Actually, this is our palace donated by the king. And we, as Central Food Technological Institute, we have a mandate that we have to work on food. In a sense, we work on this particular focus that food should be a medicine and medicine should be a food. That's said by Hippocrates. So in this angle, if you look at phytochemicals, several in terms of COVID therapy, there are several phytochemicals which has been predicted to bind to COVID and could possibly inhibit, but there's not a single report where it has actually been shown that phytochemicals could actually inhibit COVID replication, things like that. But here I would like to introduce that why not use RNA-based therapeutics instead of protein-based therapeutics like phytochemicals, because RNA gives you much better specificity. For example, if you consider this is a COVID genomic RNA or it's mRNA. If you use an antisense oligo, basically what happens is, or any antisense RNA, they'll go and bind to the target mRNA and cleave them so that you don't get the protein. So we are kind of blocking it even before the virus starts making the secondary virus. Because if you look at this picture, once the virus enters inside, you have loads of viral mRNAs, different genes coming out of it, and these RNAs are naked. Even though viral genome is kind of encapsulated by nucleic acid protein, once it goes inside, you have several targets here. So even before the secondary virus is made, even before the proteins are made, if you have RNA-based therapeutics which can bind to these viral mRNAs and then degrade them, then you can easily target this virus much before the secondary viruses are made. So you have several different types of antisense technologies like siRNAs, antisense oligos, sgRNAs, aptamers, ribosomes, and small RNAs, things like that. But here I would like to focus on microRNAs. So what are microRNAs? Basically, these are discovered about 25 years ago. These are endogenously made small RNA transcript, about 18 to 22 nucleotide in size. The beauty of microRNAs is that they regulate more than 2/3 of the genome. And they also exhibit promiscuity that one microRNA can target several mRNAs. Everything works by complementarity. And even one mRNA can be targeted by multiple microRNAs. And they are actually, some of the microRNAs are already in clinical trials. So the way they work is, when you have a microRNA, it goes and binds to the target mRNA by either with the perfect complementarity, or with the partial complementarity. Depending on its perfect or partial, the mRNA will undergo either degradation or translational repression. And the reason why I prefer binding microRNAs instead of phytochemicals is because these guys exhibit better binding energy. If you look at phytochemical papers, they will show the docking of phytochemicals with SARS-CoV-2, spike protein, all those things. You will hardly see -10, -5, that kind of binding energies. Whereas using this complementarity approach, like nucleic acid-based approach, you can actually reach more than -30 also. And they have better target specificity, and you can synthesize them in large scale, and they have better stability. You can confer better stability to them by modifying them, like phospholipid modification, locked nucleic acid modification. And specifically, plant microRNAs are better than human microRNAs because they have an extra modification, that is 2' methylation. So this is where I introduce this terminology called cross-kingdom regulation, because here we're going to talk about microRNA from a different kingdom regulating microRNA, I mean, the target mRNA in different kingdom. For example, microRNAs from plants targeting either a pathogenic mRNA in human or the viral mRNA or viral genome. This is known as cross-kingdom regulation. This is an emerging field right now. So there are some reports where they show that several food-derived microRNAs like honeysuckle, tomato, or rice-derived microRNAs, they can actually go inside you and target, regulate the genes in you. That is like, for example, 168 was shown to regulate LDL RAP1. So, but the reports are really few. One of the reasons why this is very few microRNAs have been shown to exhibit this cross-kingdom regulation is, we should imagine when we eat food, for the microRNA to pass through the GI tract and get into your bloodstream, it has to undergo a lot of hurdles. First thing is the mouth, you have the enzymes and all those things, and you have the stomach pH, and then you have loads and loads of nucleases in your intestine. And crossing all these things for a microRNA or any RNA molecule to enter into circulation, it's definitely impossible. But it doesn't mean that it doesn't happen. It actually has been shown recently for SARS-CoV-2 that there is a microRNA called microRNA 2911 from Honeysuckle decoction. It's very interesting because, in traditional Chinese medicine, they've always seen that when you drink Honeysuckle decoction, you will get rid of the inflammatory diseases, respiratory diseases, it's been shown for viral diseases, especially respiratory syndrome related viruses. And they extrapolated this further, where they show this particular microRNA from Honeysuckle decoction has several binding sites in SARS-CoV-2 genome, like about 28 different binding sites being complementary regions for this microRNA. And uniquely, this microRNA is able to get into your system. Once you drink a lot of Honeysuckle decoction, if you measure serum microRNA, you see loads of microRNA coming up in your serum. And in fact, it can actually inhibit SARS-CoV-2 replication. Either you drink a lot of Honeysuckle decoction and get your serum exosomes, or you transfect cells with the synthetic microRNA mimic, microRNA 2911, get that exosomes and incubate that exosomes with the SARS-CoV virus, in cell culture assay, you can actually decrease the amount of SARS-CoV-2 virus produced, which means this microRNA is able to get absorbed and can inhibit SARS-CoV-2 virus in vivo also, because they've shown this particular data where the people who drank lots of this Honeysuckle decoction compared to traditional other TCM methods, they have lesser positivity rates. Their RT-PCR comes from positive to negative much faster. But there are certain points to note here. The reason why this microRNA 2911 is resistant to, or it's able to absorb into systemic circulation, is because it's highly GC rich. The reason is several RNAs are, they target the AU base, and that's why it exhibits resistance to RNases. And it's been shown that it gets absorbed somehow into epithelial cells, and cells itself somehow packs them into exosomes. So it's also protected from your serum circulation, and it's released in circulation so that it goes and performs function in target organs. But majority of plant microRNAs do not exhibit this function. They are degraded in the GI tract. This is called as a bioavailability problem. It's a very famous problem in plant, phytochemicals field, in which majority of plant phytochemicals do not exhibit bioavailability, which means whatever you eat doesn't get absorbed in your system, which is also pretty true for microRNAs also. So that's where my lab focus comes into picture. I work on something called as edible nanotechnology. I would like to quote this. There's plenty of room at the bottom, like nanotechnology. So why would I talk about nanotechnology? Because exosomes are actually naturally occurring nanoparticles. They are like 60 to 100 nanometer size. And these exosomes are, they are natural nanoparticles, which is made by multivesicular bodies by your cells, and they get released. And exosomes are getting a lot of attention, even to increase the bioavailability of phytochemicals. People are using exosomes. They dump exosomes with curcumin, which doesn't have any bioavailability. You can put anything inside exosomes, hydrophobic drugs, hydrophilic drugs. So in mammalian system, exosomes mean bioavailable components. But it turns out even plants also have exosomes, because if you look at evolution, making these exosomes or extracellular vesicles is a conserved phenomenon across different evolutions, different kingdoms. For example, bacteria are known to make extracellular vesicles. Mammalian cells make it. Even viruses budding out of cells is also similar to exosomes. And it was probably about 50 years ago, almost 50 years ago. People observed carrot cells have these multivesicular bodies, which is contain these extracellular vesicles, sorry, which is ready to be secreted. It was initially thought as artifacts, but now it turns out presence of exosomes or extracellular vesicles is much more common than imagined. Here, one example is there where you just take this common grapefruit and then just fix them with formaldehyde and just take a section out of it and observe under electron microscopy. You can actually see the presence of these multivesicular bodies, extracellular vesicles in this grape. And there's an emergence of interest in these extracellular vesicles in plants, and it turns out that several plants seems to have them. For example, blueberry, ginger, you see these little small dots here. Small little, this is atomic force microscope pictures of small vesicles derived from these plants, naturally present. In the field, they are called as edible nanoparticles, but actually they are extracellular vesicles derived from these fruits, vegetables, whatever it is. But why would I be interested in this? Because they give you bioavailable, they give you the property of bioavailability. That is, they protect whatever is inside these edible nanoparticles and protect them from GI tract digestion and sort of send them into the epithelial cells. For example, in case of ginger, when you isolate edible nanoparticles from ginger, they show the key bioactives, gingerol and chagarol in these edible nanoparticles by TLC. And these ginger nanoparticles or carrot nanoparticles, grapefruit nanoparticles can get spontaneously absorbed into your intestinal epithelium. Here you see that this is the blue color labeled as intestinal epithelium, where the red colored dots are actually these nanoparticles purified from ginger and carrot and grapefruit. They can get in spontaneously and deliver the bioactives, whatever is there inside. And that's the reason why there are several papers coming up right now where they talk about, research articles which discusses the anti-inflammatory, anti-cancer activities of these nanoparticles from different fruits and vegetables, and conferring biological activity against any kind of disease, inflammatory disease, things like that. So, but the point is, and obviously these are eco-friendly, these are purified by eco-friendly protocols, and they exhibit very low toxicity, very low immunogenicity, and they exhibit very good tissue-specific targeting. That I'll come to that in a minute. And they are purified by eco-friendly protocols. There's no organic solvent involved and things like that. And they're also they have significant potential for large-scale protection, and they also do not cross the placental barrier. They are non-toxic. As I said, you can actually manipulate these edible nanoparticles to different tissues by multiple methods. For example, this is in this particular report in 2013, the grapefruit derived nanoparticles, exosome-like vesicles, depending on whether you inject them IP or intravenously, they can actually reach different tissues. These nanoparticles are actually labeled with the fluorescent dye, so you can track them in vivo. So depending on the route of administration, you can sort of push them into different organs. And if you give them intranasally, actually they can get into lungs and stay for almost 48 hours. I would like to highlight here because in SARS-CoV infection, lung is the major organ, right? So this will be very useful in that particular case. You can also target them to cancer tissues by manipulating these edible nanoparticles. Just by putting a folic acid into these nanoparticles, by incorporating folic acid, you can target them to cancer tissues, because cancer tissues takes a lot of folic acid. You can also use them as nanoparticles to incorporate chemotherapeutic drugs, so you can kind of target them to different tissues. So the take-home message is these are amenable, these are manipulatable, where you can achieve different tissues targeting by route of administration. And you can also, and it's also been shown, depending on the lipid composition from which plant you're taking it from, whether it's from grapefruit or ginger or tomato, depending on the lipid composition, they also have some tissue specificity. And you can also target cancer cells by coloring modulators. So why am I saying this? Because these edible nanoparticles also have microRNAs in bioavailable form. So the point is, if you have microRNAs in these edible nanoparticles, then they will be protected from the problem of bioavailability, where they will be protected from gastric juices and nucleases because they are encapsulated in a membrane-like vesicle. In fact, this particular paper, they show that they purify edible nanoparticles from almost close to 11 different fruits. Each one of them have about approximately 40 to 125 different kinds of microRNA with a size of about 21 to 22 nucleotide in size. And in fact, I mean, just to show you this, there's already a cross-kingdom regulation where they show that if you give ginger-derived nanoparticles to human beings, what happens is these ginger-derived nanoparticles also gets absorbed by gut bacteria through targeting some bacterial genes, like plant microRNA targeting a bacterial gene, by which it changes your gut microbiome profile to much more beneficial microbiome system compared to what it was before. So, but so far, ENP-derived microRNAs targeting virus genome or transcriptome has not been described yet. So this is where we hypothesized, okay, why not use ENP microRNAs for targeting COVID? So this is the hypothesis. This will be an excellent alternative and a faster therapeutic option because if you have microRNAs targeting COVID, you can actually target these ENPs by intranasal administration. We try to get into the lung, and even if you do not have COVID, this is non-toxic. It's going to stay only for 48 to 72 hours, so that the microRNA is already there. So even if you get COVID infected, they will take care of it. So the point is, so we take the plants, isolate edible nanoparticles, intranasally deliver them, so that you have these nanoparticles fuses with the lung epithelial cells. So you have the microRNA, and then when you have SARS-CoV, in case you have SARS-CoV infection, then these microRNAs, which is specific for COVID-2, when SARS-CoV-2 genome or transcriptome, we bind and inhibit the replication. But do plant edible nanoparticles have COVID targeting microRNAs or not? That's the crux of this particular discussion here. So what we did was, there are only 3 studies so far in which they have profiled microRNAs from about 12 or 13 different fruits and vegetables. So what we did was we got all the top 20 microRNAs from each study, because each plant has about 40 to 120 microRNAs, but it's been shown that top 20 microRNAs constitute more than 90% of the total microRNA counts. So we ended up with about 260 microRNA sequences, and some of them are partial because of sequencing errors and things like that. So we retrieved the full-length microRNA sequence, and we used a very stringent selection criteria, which I'll describe in a minute. And with this, and did a target prediction. So we got about 22 microRNAs that could potentially target COVID genome, and we checked the specificity, target site accessibility, and we also validated some microRNAs in ginger ENPs and grapefruit-derived nanoparticles. So this is the criteria we kept. That is, the first thing, to achieve really high specificity and sensitivity, we said we want only those microRNAs which can bind to COVID genome or transcriptome with more than minus 25 kilocalories per mole. And we are talking about cross-kingdom regulation. As I said, the way microRNA works in plant is different, where we need complete complementarity, whereas the way microRNA work in animals is kind of different, where partial complementarity would do. So we allowed GU-based pairing. This is a GU-based pairing here. So instead of having GC, you can also sometimes have GU. But we place helix constraint between 2 to 8 bases. When I say helix constraint, that means that I need to have complete complementarity between 2 to 8 bases, because in animals, this is called a seed sequence. So which means we need to have perfect complementarity. You can have some kind of bulges, this kind of bulges in between, but you need to have complete complementarity in seed sequence. So we kept that here. We also allowed some internal bulges. So with this criteria, we use RNA hybrid software to predict what are the microRNA, out of 260 microRNAs, how many of them bind to SARS-CoV-2. And we could actually get almost close to 22 microRNAs, which could bind to different genes in the SARS-CoV-2, like from spike to ORF8, ORF1AB, and multiple almost 22 microRNAs targeting multiple genes, all of them exhibiting more than minus 25 kilocalories per mole energy. So this is how it looks like. So you can see that this is the bind. It's like phytochemical binding to the target protein. But here it's much more simpler, where it's just looking at hybrid structures between your microRNA and the SARS-CoV-2 region. So the green color is the microRNA, and red color is SARS-CoV-2 region. You can actually see very nice complementarity sequence binding between 22 different microRNAs in the target regions, and a bit of bulges are there. Okay. The second thing is, we also looked at whether these microRNAs which target SARS-CoV-2, the binding region, is it conserved Is it conserved since this is specific for SARS-CoV-2, or could it also bind to SARS-CoV, which is a 2002 outbreak of coronavirus? So what we did was we took the binding sequence and compared it with SARS-CoV, the 2002 outbreak SARS-CoV. So we took these different strains of SARS-CoV-2 along with SARS-CoV-1, the previous one, and looked at what is the conservation of the binding site. As you can see here, the 3' end, you have some mismatches right here. So these mismatches tell you, as I said, the seed sequence is pretty important. This mismatch is present only in SARS-CoV, but not in SARS-CoV-2, which means these binding sites or the microRNAs targeting these binding sites would be specific to SARS-CoV-2. They will not bind and execute their function to SARS-CoV. Whereas we also had some microRNAs which showed complete conservation, which means these, the microRNA 530B, 535B, slime, 1919E, these microRNAs can also bind to SARS-CoV as well as SARS-CoV-2. We had some microRNAs which had seed sequence binding, but pretty good other mismatches in the other areas of the it's like this kind of gaps were there. It's difficult to predict whether it's going to bind to both or not at this point of time, because it also depends on the intracellular context. So this may or may not bind to both genomes or transcriptomes. So having the microRNA binding site alone, it doesn't necessarily mean microRNA is going to bind. The point is whether the microRNA binding site in the genome, is it accessible, whether there are any secondary structures there. So there are two ways by which you can calculate this. First thing is, what if the microRNA binding site itself, because RNA is bound to form secondary structures, it's always forming the stem loop and stuff like that. So we have to make sure the regions where the microRNA binds do not have much of secondary structures, and also the regions flanking the microRNA binding site also should. There are certain rules and regulations here. If they have second structures, then microRNA binding will be affected. So we check both of them by subjecting the target binding sequence along with 12 to 17 bases upstream and downstream sequences to Mfold analysis and see what are the second structures that is there already. So only out of 22 binding sites, we had only about 2 of them showed secondary structures, which is more than minus 15 kilocals, like minus 20 or minus 17.5 for this binding sites. But majority of them are less than minus 15, so that since we should remember that our microRNA binding site is binding energy is almost close to minus 25, so they will be able to, they'll be dominant in dislocating the previously existing structures and go and bind to them. So that way it will be much more efficient. And we just compare the expression profiles of these microRNAs based on the NGS data analysis. As you can see here, so basically what we did was these are the COVID targeting microRNAs, and the relative expression levels is plotted as a heat map here in different ENPs, different nanoparticles isolated from different fruits. As you can see here, you take tomato, isolate ENPs, that would have at least 4 microRNAs which could potentially target COVID-2 with very high expression, like more than 1,000 copies, more than 100,000 copies. And in addition to that, you also have another 3 microRNAs which is expressed, which is present in lower amount. And likewise, you have grapefruit, likewise you have soybean, which has 3 microRNAs. So obviously, based on this, we can sort out which ENP is best for SARS-CoV-2 targeting. So lastly, what we also did was we want to verify that, because this published data from other countries, we want to verify whether is it universally true, because obviously any plant you take, they show local variation. Like a ginger from China, a ginger from India, and ginger from the US, it's always different. So they may not have the same profile. So we actually purified the edible nanoparticles from ginger and grapefruit. And so usually when you purify these nanoparticles, they'll be approximately about 400 nanometer size is the size of these nanoparticles. That's what is shown here. And they also show us Zeta potential of about minus 25, minus 30 millivolts, suggesting that they have negative charge vesicles and they're kind of stable. They repel each other. So that minus 25, minus 28 millivolts mean they will repel each other. So they will not aggregate in future. So when we purify small RNA population from these, so what we did was we purified the small RNA population and first loaded them on gel. You can see this kind of small RNA population here. This is 100 base pair, double-stranded DNA. This is 500 base pair. So you get much lower small RNA population. Just to confirm, this is RNA. We also loaded RNase A here, so that you can actually see the disappearance of these bands, suggesting that these are indeed RNAs. And we subjected them to qPCR for specific microRNAs. So microRNA 166M and 6300 are microRNAs which is present in equal amounts between ginger and grapefruit. So you can actually see the same thing here. These are raw CT values which is plotted. Actually, you can get sometimes get CT values as high as 10, like which means it's really abundant microRNAs. And this particular microRNA 159 and 5059 and 5077, they are highly expressed in grapefruit. They are much higher in grapefruit compared to ginger, whereas this microRNA 156 is much higher in ginger compared to grapefruit. So the point is that we have identified about 22 different COVID targeting microRNAs in our study. And these microRNAs target both structural as well as non-structural genes. And each plant contains, like honeymelon, soybean, tomato, pear, ginger, grapefruit. They contain multiple microRNAs targeting SARS-CoV-2. So it's like one bullet but multiple targets. And so there are some microRNAs which is present in everything. For example, microRNA 6166M, which is present in every plant, whatever you are, in every ENP, whatever you isolated from. As I said before, ENPs, when you give them intranasally, they get into the lungs. So which means that you just make a nasal formulation of this kind of nasal spray out of this ENPs, and you spray it to the nose so that they go and fuse with the lung epithelial cells, stay back there, so that when you get an infection, or if you're already infected, these microRNAs can get into these cells, and then they could potentially suppress the replication of SARS-CoV-2. I know it's a kind of long shot, but these has to be experimentally validated with the chimeric luciferase assays or in vitro cultures. But since we didn't have those in vitro culture facility, we couldn't do all those things. But as a take-home message, I believe this ENP-derived microRNAs itself is like a treasure trove, which can be used for any disease, not only COVID, any other viral infections. We can predict them, we can validate them, like or pathogenic, for example, adipogenicity. You have to inflammation markers. We can use these microRNAs to target these inflammatory markers. So they have a lot of potential. So this has been published here in Toxicology and Applied Pharmacology recently. So we'll be happy to. Before that, I would like to acknowledge the person who did all this work. This is my student, Mr. Sriram. So this is the lab we work here at CFTRA. So thank you for your attention. I'll be happy to take any questions.